All right, welcome everyone. Sorry for the slight delay in starting class. I hope you're having a beautiful, sunny Monday. At least parts of it have been sunny. Let's get to it, shall we? So we are on our final week. Today, we're going to talk about the innate immune system. Tomorrow, we're going to talk about the adaptive immune system. This part of the immune system, as you are certainly aware because you've taken it before, is the so-called non-specific defenses. However, it is really important to understand that that's kind of an oversimplification of the case. So when we think about what our two branches are, the innate and the adaptive, there's multiple differences between them actually that go beyond this specificity issue. Certainly it is true the adaptive immune response is much more specific, but it's not that the innate immune system is totally blind. So while it's non-specific, it has the ability to recognize patterns, especially bacterial molecular signatures. And also just focusing on the specificity kind of overlooks this other really important part that's different, which is that the innate immune response doesn't change or adapt to the environment, whereas the adaptive does. So the adaptive immune system actually starts out early on as being not very useful, and then it gets better and better by being exposed to the world. The innate immune response is really helpful right out of the gate for just protecting you generically against dangerous things, against bacterial infection. Although it doesn't really get substantially better. In fact, it eventually gets worse because your body starts to fall apart and everything falls apart. The other thing that's really important to understand that's sort of obscured by this simplistic distinction between these two branches is that they talk to each other. They lean on each other, they collaborate, and we're going to see lots of examples of that. So when we're talking about our subject today, we also want to understand how relevant all of the stuff we've talked about before is. And so in particular, in this section, we talked about portals of entry in epidemiology and in pathogenesis. What were some of those portals of entry? If you don't remember anything, put a question mark into the chat. But if you can remember one of the portals of entry, even just go ahead and put it in. Yes, that's right. Skin and mucous membranes are the primary portals of entry. Then there's also this other one, parenteral. Anyone remember what this one is? That's right. So parenteral is when the skin is broken, a wound, a cut, or even a needle stick, right? Now, what is it that is important about portals of entry? Where in epidemiology, we cared about portals of entry and portals of exit because of the spread and transmission of disease. Why do we care about it now in immunology? What is the relevance of portals of entry to our current subject? Yes, yes, exactly. This is our first point of contact, right, with our pathogens. So it's certainly going to be a place where we put at least some of our defenses. Now, in pathogenesis, we focused on the molecular toolkit of pathogens, how they invade us, how they move within us, and how they make us sick. Those virulence factors are relevant again today. But now from the point of view of immunology, we care about them because they tell us what we are up against, and it informs our understanding of the defenses that we have, what defenses we should have, what defenses we do have, and how effective they might be. I'd like us to recall some of those virulence factors. What were some of the things that allow pathogens to bind to our tissues? Anything you can think of, put that into the chat. What were some of the virulence factors, the the tools that pathogens allow or use in order to grab onto us. Yeah, bacterial fimbriae and viral spikes were two good examples. Many microbes have some way to avoid phagocytosis. Can you think of any that we have covered? Yes, bacterial capsules. Yes, Janelle, the ability to mutate. And of course, as some of you mentioned earlier, one of the virulence factors that's relevant here is flagella, the ability to get away. Additionally, virulence factors include things that cause damage. So what might those things be? Yeah, endotoxin, that's one. Besides endotoxin, are there other ones you can think of? Yeah, exotoxins also. Not to mention things like exoenzymes or metabolic wastes. So in these two branches of the immune system, there's actually three lines of defense, but the first and second fall under the auspices of the innate immune response, which is to say you get them right away and they don't really change. So let's review what's in them. First, let's sort of think metaphorically, or what would be a good analogy? What is that first line of defense like? Can you think of something in the real world to compare it to? Yeah, a fence, a gate, a wall around a castle. I think of it as a wall around a city, a wall around a castle. It's good too. The guarded entrances would be the mucous membranes and the wall would be your skin, right? So in addition to skin and mucous membranes, what else is in the first line of defense? Oh, I like that, a glass protector. That's a good one, Dorothy. Besides skin and mucous membranes, what else is a part of our first line of defense? That's right, Haley, normal microbiota, correct. All right, so now we're going to review how each of them protects us. This is from your prep. The skin is dry, which most microbes require moisture. Acidic, most microbes are neutrophils. Multi-layered, and it sloughs off. Very waterproof as well. Mucous membranes have mucus, often cilia. Antimicrobial substances like lysozyme, and normal microbiota, of course, occupy and protect. So if we have a quick look at all of these, the physical barrier of the skin, these multiple layers of dying skin cells provide a waterproof barrier, and it's very, very thick and difficult to penetrate. It's dry, it's acidic, and it repeatedly sloughs off, taking the surface microbes with it. While mucous membranes are not as well protected, they're tall and sometimes they have cilia. They always have goblet cells making mucus that traps microbes, and the cilia, when it's present, can move them along. Particularly important in the lungs, right? Or in the respiratory tract, I should say. The ciliary escalator moves the mucus up in opposing the direction of gravity, moving the microbes out and out of your respiratory tract, protecting the lower airway. And then we've got the muscular contractions of your GI tract, the secretions from the genitourinary tract, and the lacrimal apparatus that washes the eye. Additionally, in your first line of defense are some antimicrobial secretions in that mucus and in other secreted fluids, like in your sweat. There's an anti-fungal component, and your sweat is salty, providing a salty surface for your skin. Most organisms that are pathogenic are not going to be very happy on your salty skin. There are some exceptions that include our normal microbiota. What genus is famously halotolerant that's found often on your skin? Yeah, that's right, Staphylococcus. It's not that it's the only species or the only genus containing species that can survive here. There are others, but it's a common resident of human skin. And the acidity of different parts of the body are helpful. So our skin is acidic, but also our very acidic, our stomach. This is for two reasons. If you've taken nutrition, you'll know this, or A&P probably, that the acidity of your stomach is in part and importantly to relax the proteins, denature them, make it possible to chop them into bits during digestion. But it's also an important way to kill off most pathogens, though there are some that can survive in this acidic environment. Most of them are going to be killed. And other parts of the body too have a low pH. Furthermore, there are a lot of bodily fluids like sweat, tears, saliva, urine, as some examples, that have lysozyme. Lysozyme. What is this enzyme? What does it actually target? What specific molecule does this degrade? That's right. This hydrolyzes the bond between NAG and NAM, which is the two sugars that form the subunit disaccharide of the peptidoglycan filament. So basically, this is an antibacterial enzyme that'll chop up the cell wall. And you'll remember that we heard it before when we talked about the bacteriophage life cycle. And then in addition to that, we have transferrin in our saliva and other places. What does transferrin do? This is something we make, and what's it for? What is its function? It binds iron, that's right. So it's binding iron to keep it away from us, make sure there's not free iron floating around, so that's reactive and damaging, but also to keep it away from bacteria. We need it so we can run our respiratory, cellular respiration. Bacteria can use it for the same thing. So just a brief reminder about lysozyme, making sure we understand this is not the same thing as lysosome. This is not referring to the organelle that's digestive. This is an enzyme. And this enzyme is something we actually share in common with bacteriophage. Right? Bacteriophage have this weapon, which they use to cut a hole in the cell wall of bacteria in order to propagate themselves. We have the same weapon to protect ourselves from bacteria. And again, what it does is it degrades peptidoglycan, and it's found in a lot of our bodily fluids, our tissue fluids, our blood, saliva, tears, mucus, sweat. Transferrin binds iron. This protects you from toxicity from free iron, making sure you keep iron and preventing bacteria from getting access to your iron. And last but not least, we have in our first line of defense the normal microbiota, and they are useful to competitively exclude pathogens. How do they do this exactly? If you can think of anything that you remember about specifically how they protect you, put it into the chat. Yes, they prevent bacteria from colonizing because they're sitting in the spaces. There's no more space. And to be more sort of like detailed about what that really looks like, they're sitting on top of your binding sites. So In order to colonize you effectively, a pathogen needs to grab you first. Well, if you're covered in microbes and all the binding sites are taken, that's not going to happen. They consume the nutrients, right? So there's limiting amount of nutrients. The existing resident microbes are eating it all, so the pathogens don't get any. Those normal microbes are also secreting metabolic waste and sometimes actively secreting weapons into their environment to keep out interlopers. Now, some of the members of our microbiota include so-called commensal organisms. We talked about this in pathogenesis. This is an example of an organism that benefits from living on us, but doesn't really provide any benefit to us, but we're not hurt by it. It's not a parasitic relationship. And these guys can be opportunistic pathogens. So they're fine in one spot. They may cause damage in another one if they go to a place that you're not supposed to have microbes on your body. Furthermore, we want to remind ourselves of the effect of antibiotics. By depleting our normal microbiota population, they can increase the risk of pathogens taking hold. That's how effective normal microbiota are in protecting you. So that's all the first line of defense, right? Let's talk about the second line of defense. A key component is phagocytes. There's also processes and so-called antimicrobial substances. So the goals of the second line, whereas the first line of defense is basically a physical barrier, the goals of the second line are more pattern recognition. They protect you if the wall's already been breached. So the bad guy got in through the castle wall. Now there's roving beat cops, there's booby traps that are set up to go off and capture and remove guys that have gotten in past the initial physical barriers. So the patterns that are recognized by the second line of defense have to do with usually molecular signatures that are most of the time that are bacterial, though there are some other ones as well. They're molecules indicating tissue damage. So when tissue is damaged for any reason, this causes so an alarm to go off and to initiate inflammation. And we're going to sort of see some of the details here. Now, a lot of what goes on, especially when we want to talk about phagocytes, has to do with our blood cells. A lot of our blood cells are involved in immune functions. Here's a list of our blood cells. There's a couple that are not. Red blood cells' primary objective, of course, is to deliver oxygen for respiration. Nothing really to do directly with immunity. Platelets are there to clot the blood, okay? But everybody else, all these white blood cells are involved in immune function. So macrophages, dendritics, and neutrophils are all phagocytes. And T's and B's are part of your adaptive immune response. We'll focus more in on tomorrow. One of the things that's really neat about the white blood cells is that there are multiple different types, and probably you've heard of these different types in a previous class. But it's not just the case that these all have different functions. They also appear different under the microscope, a light microscope, not even an oil objective, but under just like a 4DX objective. You can, with a little bit of training, tell them apart. And so when you do an analysis of the white blood cell composition of a normal, healthy person, and you categorize, let's say, the first 100 white blood cells that you see and put them into these different categories, then you see a pretty predictable range or ratio of these guys. You see the most of them are neutrophils, somewhere between 2/3 to almost 3/4. Very few of them are basophils. A small number are eosinophils or monocytes, and then the rest are between 1/4 to 1/5 of them are lymphocytes, the B's and T's. So this is what it looks like in a normal person. Now, the really helpful thing is that just by quickly analyzing this, you can tell if the ratios are off. And that could even tell you not just that someone might be ill, but even give you a clue as to what kind of illness they might have. So when we see a disproportionately large number of neutrophils, what does that mean? That's right. Neutrophils are so-called first responders. They're phagocytes, but they're first responders. There's lots of them, and so they're the first to the scene usually. And if we see more of them than normal, what it usually means is that we are early in an infection. Basophils are histamine secreting cells, and they're pretty rare. If we start to see more of these, what could that indicate? Yeah. Inflammation, especially relating to allergies. Eosinophils have a very particular role. When we see more than we are expecting of eosinophils, what does that often indicate? Yeah, eosinophils are antiparasitic. So seeing more of these suggests that someone has a parasitic infection, although it could also mean that they're dealing with chronic allergies. Monocytes are the precursor to macrophages. So a higher level of these typically means that we are in a later stage of an infection or in an infection that's not clearing up. And last but not least, if we see more lymphocytes, T's and B's, than we would normally see, that usually means our adaptive immune response has kicked in. Whereas on the other hand, if we see a reduction in numbers of lymphocytes, that could indicate that our immune system is failing. And this is what we see, for example, in the final stages of an HIV infection. The T cell count goes down. Okay, let me make sure that I have my sound on when I share this clip with you. All right, here we go. That's just all kinds of fun every time. Let me ask you some questions. First of all, where are we? Yeah, this is a blood sample. And all these like round guys that are all over the place, what are these? That's right. Yeah, good guess. Those are red blood cells. And that would make this bigger one over here. What's this? This like lumpy one over here. Yeah, these are white. This is a white blood cell. We even can say for sure that it is a phagocyte. And if you remember reading the title, then you probably know what kind of white blood cell it is. But even if you didn't, you could take a stab at it. What was the most common type of white blood cell? Neutrophil. That's right. This is a neutrophil. Now it's chasing something. Can you determine from this video what it's chasing? What category of thing it's chasing? Is this a parasite? Is this a virus? Is it a bacterium? Like, what is this? Well, let's wait, and I'll just let everyone weigh in. So this little dark thing down here is what's being chased by this white blood cell. Lock in your votes, guys, because that's more fun. And thank you to everyone who's answered so far. If you have no clue at all, you're always allowed to put a question mark in, but do make a stab at it. Great. So yes, bacteria is the right answer. Raise your hand if you can explain why. What tipped you off to that? How did you know? Other than it said so in the title, but most of us didn't pay any attention to that. What, if you had not seen the title, how would you know this was bacteria? What are the clues? Raise your hand. Or how do you know it's not a virus, not a parasite? Yeah, Haley says, could it be due to its size? Yeah, its size is a good clue. It certainly doesn't appear to be eukaryotic. Red blood cells are among the smaller eukaryotic cells, and this is much smaller than that. A parasite would be roughly the size of this, this white blood cell usually, right? So it's not a parasite. It's not a virus, because if it was a virus, this is a light microscope, right? It's a time lapse, but it's a light microscope, and you wouldn't be able to see it at all. The other thing that's very telling, given its, small size ruling out eukaryotes, but now we're down to, well, it's something smaller than that, is it's moving. It's running away, which viruses are not able to do. So even if this was an unusually large virus, we would know it's not a virus because it can run. Okay. All right, so there are 3 types of phagocytes, as we mentioned earlier. List them in the chat. Or even if you just remember the name of one type of phagocyte listed in the chat. There are 3 specific ones. They have different jobs to do, right? Which makes sense, because otherwise, why have 3 types? Yes, macrophages, neutrophils, dendritic cells. Let's start with the dendritic cells. What is their, it's a phagocyte, so are the others, but what is sort of unique about the dendritic cell? What is its primary function or role in the immune system? How would you describe that? What's distinctive about it, what it's doing? Okay, yeah, it's part of the alarm or alert system to alert the lymphocytes, that is to say, coordinating with T cells, right? Its job is to be a scout, to go out and find stuff, not to clear it. Its job is not primarily to clear the infection, but rather to tell the T cells what's out there. Okay, so another type that we had was the neutrophils, and those are our so-called first responders. They're very frequent, they're enriched, or they're at a high percentage of white blood cells, and they don't live very long, so they're patrolling your body all the time. They're your first responders, like the fire and EMS and police, right? First responders, whereas the macrophages, which are much more aggressive, need to be activated, are more like deployed troops, right? You bring them in once you're like, okay, now we know we're under attack. So we have our first responders, we have our scouts to talk to the adaptive immune response, and we've got our deployed troops. These guys will be called in once we already know we're dealing with an infection. Okay, now phagocytosis, whichever one of those 3 is doing it, follows a series of steps. So what are those steps? What I'm going to have you do, don't put anything in the chat yet, just think about it. How would you describe the process of phagocytosis from beginning to end? I'm going to cold call people. And what I'm going to ask you is, what happens first? And then when we get to what those events are, I'll ask another person what happens next, and we'll get to the end of phagocytosis in this way. So think about how you would describe the first, second, third, fourth step in phagocytosis, if we were to break it down. And then I'll ask someone to give me the first step. And you know my rule. If you don't have an answer, you can always pass. It's not a problem. But let's begin with Dorothy. what is the first step in phagocytosis? What do we have to happen first?
Appearance.
That's right. We have to find and bind or adhere to our target. So that's the first step, is adherence. Good. Let's go to Janelle. What happens next after we have found and bound to, grabbed onto our target? What does the phagocyte do next?
Engulf.
That's right. Brings it inside, endocytoses it. And that creates what, guys? Write this into the chat. Once you have engulfed it, it creates something called a what? What's that vesicle that's full of whatever you've captured called? That's right, Haley, it is called a phagosome. Okay, so now we have engulfed our target and it's in a little vesicle. Ayumi, what has to happen after that? What's the next step? We have our phagosome. What do we do next?
Digest.
Before digestion, but you're on the right track. What has to happen before we can digest? You're absolutely on the right track. We just want to make sure we kind of get down at the details here. If you're not sure, you can say so, but if you can think of something, let me know. What do we need in order to do that digestion?
I'm sure, but on my notebook, oxidative burst.
Yes. Okay.
Yeah.
Let's see. Yeah, no, you're absolutely on the right track. So let's see if someone else wants to add to that. Lori, do you have anything you want to add? Like right after we have created the phagosome, in order to accomplish the digestion step, what do we have to do?
Digest. Digestion.
Yes. And what's going to help us with that? Digestion is not just going to happen spontaneously. We have engulfed our guy. Can you think of what we might need in order for digestion to occur? If you're not sure, it's fine to say so, but go ahead.
Is it enzymes that we need?
We do need enzymes. And it looks like Tao has raised her hand and wants to contribute as well. Go ahead, Tao. How would you like to help out? You have your hand up. Tao, go ahead. You are still muted. Okay. Oh, she put it in the chat. Thank you. It says fuse with a lysosome. Yes, your microphone is not working. That's fine. Fuse with a lysosome to create a phagolysosome. So we need the organelle called a lysosome, which is the digestive organelle that contains those enzymes everyone's talking about, those digestive enzymes. We need that lysosome to fuse with the phagosome, creating a phagolysosome. So now the enzymes are inside the phagosome, which is now a phagolysosome, and digestion can occur. And last, once digestion is completed, raise your hand if you know what the last step is. Sarah, go ahead.
Discharging the waste material.
That's right. We have some undigested material, an indigestible material, kind of like fiber in your diet. It's kind of like that, I suppose, that gets expelled by the cell. So this is a nice little diagram that summarizes those steps. We have the finding and binding. Now, there's lots more we could say about that, about the specific receptors and molecules and things that are involved in adherence, but let's just leave it at finding and binding. Engulfment, creating a phagosome. Lysosomes fuse with the phagosome to create a phagolysosome. So now we have digestion. Some of the things can't be digested and they're spit out in the process of exocytosis. Now, are phagocytes always able to catch their man like in that video? Not always. So what are some of the tools, strategies, defenses that bacteria have to avoid phagocytosis? Put that into the chat. Capsules, that's right. Remember we talked about how capsules make bacteria invisible, and there's also capsules around some fungi. Anything else? M protein. Yes, some components of the cell wall, for example, M protein that we learned about in pathogenesis, are also good at preventing that binding event in the beginning. Anything else come to mind? Anything that might help bacteria to run away or to survive even if they are engulfed? Yes, so flagella can help them to run away, as you saw in that video, though in the end it was not successful. And yes, the ability to mutate and hide in that way. Here is a list of some of these microbial strategies of evasion. And you are not required to memorize the ones on the right. Okay, these are just examples to give you a sense of the diversity of these techniques. It's not that all of these are employed by all species. Different species have different strategies, but some microbes have the ability to avoid being found or avoid being bound. Others will just take a stand. They'll stand their ground and they'll fire back using poisons, leukocidins that explode white blood cells. And there's several ways you can do this. Still others will, even though they are engulfed and a phagosome is formed, they escape from the phagosome. And this includes species that use that technique we talked about, where they have a chemical that mobilizes actin filaments to provide propulsion. So they can kind of explode out of the phagosome and even out of the cell. And still others do get found, they get engulfed, they form the phagosome, but they prevent the lysosome from fusing to the phagosome, so you never can get digestion. And that includes the causative agent of tuberculosis, which is one of the reasons it's such a hard infection to clear. And then I think by far the creepiest, scariest dudes are the ones that get found, they get engulfed, they get exposed to lysosomal enzymes, and they don't die. So all of these are possible, depends on the species. Okay, now, in order for phagocytes to do their job, they have to actually be in the right place. So how do they migrate to where they're needed? We have to talk about that a little bit. So hold on one second. I'm just going to share a different screen with you. Make sure the sound is on. And let's watch this very useful video.
The white blood cells are an important part of our body's immune system. Neutrophils are a special group of white blood cells that play an extremely important role in protecting the body against infections. Mature neutrophils do not divide. New neutrophils are constantly formed in the bone marrow where they develop and mature. Mature neutrophils are released into the blood where they circulate for 3 to 12 hours and then move to other tissues where they survive only 2 to 3 days. These mature neutrophils can be found in large and small blood vessels, as well as in the tiny capillaries in different tissues. In the bloodstream, neutrophils act as surveillance cells searching for infections. Neutrophils can sense a site of infection because of chemicals that are released by the bacteria. Upon detecting these chemicals, neutrophils slow down and begin to stick to the walls of the blood vessel. They then squeeze between the cells of the blood vessel wall and enter the infected tissue. Here, the neutrophils crawl towards the infection as they continue following the chemical signals. At the site of infection, bacteria are coated with antibodies of the immune system and marked for ingestion. The neutrophil recognizes the marked bacterium, engulfs it into its body, and traps the bacterium in a sac called a phagosome. The neutrophil contains pouches called lysosomes, which are full of digestive enzymes and chemicals that can kill bacteria. The pouches, or lysosomes, then combine with the phagosomes containing the bacteria and release the enzymes and chemicals, resulting in the ultimate death of the bacterium inside the neutrophil. After killing and digesting the bacteria, the neutrophil is spent. Having completed its duty, the neutrophil shrinks in size and breaks up into smaller pieces that can be recognized and eaten by other cells.
Okay. So I think that video nicely illustrates the steps in phagocytosis as well as in the steps of migration. As far as migration, you saw that the phagocytes begin often circulating in the blood, and they're just waiting for signals from tissues that are in trouble. And then once they receive those tissues, they start a process of margination. What does that mean? Put that into the chat. How would you define margination? Yeah, that's right, Amy. So what they do is they stop going sort of, they're no longer in the main flow, like rushing through the vasculature. They start to migrate to the edges and roll along the inside of the blood vessel. That gives them a chance to slow down, follow chemical signals, right? Follow chemical signals to where the trouble is. And then they can initiate diapedesis. And what does this mean? They're inside the blood vessel. They got some signals. They're starting to marginate, slow down, follow chemical signals that'll tell them where the tissue is that's nearby. And how are they going to get there? Yeah, they're going to squeeze out in between the endothelial cells that are making up the blood vessel in order to get into the adjacent tissue. This is amazing. So they make themselves really skinny and flat, and they squeeze out into the tissue. They follow more chemical signals until they find their targets, which are usually coded with antibodies, right? And then they do what they need to do. Let's have a look at our time. I think we can take our break now. Let's do that. We'll talk more about the process of inflammation, but let's start again at 6 o'clock. So I will see you then. I'll see you at 6. Have a good 10 minutes. All right. So phagocytes participate in one of the most important processes in innate immunity, which is inflammation. And this is caused by some kind of damage. It could be an obvious superficial wound, like a puncture, but it could also be internal damage. This could be from an injury that doesn't break the skin, but still causes, damage to the blood vasculature or to the tissues, and the tissue damage will initiate inflammation. But it could also be something more subtle. As we age, we typically start to get damage on the inside of our vasculature, our blood vessels, and that can be from, higher levels of blood sugar causing reactive damage to the molecules lining the cells, which can lead to accumulation of lipids and cholesterol plaques, and that causes a chronic inflammation inside the blood vessels. So it can be all kinds of things that initiate inflammation, but somehow the tissue is damaged, and that sends signals that recruits the arrival of macrophages, neutrophils, and other phagocytes. It also recruits lymphocytes so that the adaptive immune response can start get a handle on what's going on. Blood loss is mediated by clotting factors that prevent bleeding out and also help to try at least to prevent the spread of invading microbes. And then as neutrophils do their job, remember from that video, they die, and then tissue is being damaged as well. So those together, this sort of dead tissue is what you see when you see like pus coming out of a wound or from an infected surface. Now, the ideal really is that you have acute inflammation, which has a rapid onset, and then it resolves and you heal. The not so good outcome is if it's chronic. And this can happen if, for example, the damage is not healable, right? So we can have chronic inflammation from damage to our arteries, or if the infection can't be cleared. One of the hallmarks of inflammation is a vasodilation. That means the blood vessels get larger in diameter. Why is this helpful? What's the purpose of this? With respect to our innate immunity doing its job, what does this permit or encourage or support? I would encourage you to think about the last thing that we talked about. Yes, Kelly is absolutely right. By increasing the diameter of the blood vessels, you're actually increasing the space in between the endothelial cells that make up the blood vessel, making it easier for diapedesis to occur, making it easier for white blood cells to squeeze out into adjacent tissues. Now, in terms of the hallmark effects, things that you can observe, measure, and record, what are some of the ways you know that inflammation is happening? Maybe visual cues or experienced things. Yeah, the area, if you can see it, if it's on your skin, it will look red. The tissue becomes swollen because fluid is accumulating in the tissue. And it can hurt. The nerves in the area get triggered and it gets hot usually. Now, some of you mentioned fever. That's not an inflammation hallmark, but it is a complementary separate process that's distinct from inflammation that we'll talk about next. So the difference is that inflammation is like a local swelling and heat and redness. Fever is a systemic increase in body temperature. Now, by increasing our body temperature, we can potentially, hopefully, put the pathogens we're trying to fight at a bit of a disadvantage. For example, that high temperature might be making it harder for them to do metabolism or for their structures to hold together. Of course, that's true for us too. What are some of the other advantages of increasing your body temperature? From the prep, what do you think might be helpful about having a fever? Yes, Haley is right. One of the things it does is it increases the effectiveness or efficiency of transferrin, the iron binding proteins, okay? It also increases the effectiveness of interleukins, such as interleukin 1. These are chemical signals that are exchanged between immune cells, so they're basically able to better talk to each other. And also stimulates interferon production, which we'll talk about later. Let's focus on transferrins for a moment. What is it that bacteria make in order to bind iron? We make transferrin. What do bacteria make? They're going fishing for iron in your body with what? This is one of these virulence factors we talked about in pathogenesis. And my last hint to you is it starts with the letter S. So do you guys remember talking about siderophores? Siderophores are what bacteria make to bind to iron. In a competition, siderophores if they were pulling on iron and transferins were pulling on iron, let's suppose it was a little molecular tug of war, well, the siderophores are going to win. They're just better. By increasing the body temperature so that transferins work better, we start to level the playing field a little bit. Now, there's clear disadvantages to fever. Just as it can disrupt the protein structures of your enemies, it can also disrupt your own protein structures. And there are consequences for this. So can you think of any of the disadvantages at sort of the organ and system level? Yeah, it changes your pH. It makes your body pH more acidic. You can become dehydrated. It affects your heart. And ultimately, a prolonged high fever can kill you. So it's a bit delicate, isn't it? Now, another part of the second line of defense, and an important one, is antimicrobial substances dissolved in your fluids. And we've talked about several of them, but I want to focus on two. And most of it will be focused on the complement, a very powerful and important component of your innate immune response. And also we'll discuss interferons. This is a really helpful graphic for summarizing and studying the complement system. Complement system is a set of defense mechanisms that can be initiated. And there's a mechanism composed of multiple proteins, a mechanism known as the core cascade that does the job. And then there's specific things that happen if complement is activated, and there's specific ways to activate the complement. Let's start with this. Why is this system called the complement, meaning to assist, not complement meaning to flatter? Why is it called the complement? What is it complementing? Yeah, that's right, Kelly. This system, though it's part of your innate immune response, and you have it, and it can be, triggered, and when it is, it just does the things it does. So it's not adaptive in that way. Nonetheless, there is a conversation possible between the adaptive immune response. The adaptive immune response can initiate the complement. So once the adaptive immune response knows what it's dealing with, it can tell the complement to go. Now, the complement system is actually composed of 9 proteins that are dissolved in your bodily fluids. And they're always present, but are they always active, or are they typically inactive? Correct. That's right. So these are circulating all the time, but they are typically not activated, which is good news, actually, because that would be a problem. And so there's a variety of activating events, but they're typically inactive unless they've been activated. Now, I like to sort of think of the complement as like a light switch, where the light switch is wired to, say, like a light and a fan and something else happens. When you turn on the switch, all the things happen. And it seems really simple, right? You just flick the switch and boom, the things come on. But actually, if you know more about it, a light switch isn't that simple. There's a plastic bit that you flick, but there's also a circuit and some wiring. So there's a series of events that happen very quickly and always, but nonetheless, they do happen. And that's the so-called central or core cascade. There's a variety of proteins involved here that once this cascade of events is triggered, when the first domino falls, it leans into all the other ones, and then these stereotypical outcomes occur. So here are all the proteins that are involved in that central cascade, and we're going to talk about them in more detail in a second. But basically, the first protein becomes activated by some trigger. You click the light switch on. That causes this protein to separate into 2 pieces, which are now active, that each have distinct functions, one of which is to activate a subsequent protein, which is then chopped in half and goes on to do various things. Now, how do we turn the light switch on? Well, there are 3 major pathways by which we can activate the complement. And what are these pathways called? Put that into the chat. So there's the so-called classical pathway, which was the first one discovered. There's the alternative pathway, because they found out there was another way and they were like, oh, well, I guess that's the alternative. And then there's the lectin pathway, because they found a third one and by now they were out of alternatives, so they had to name it after something actually useful. Any of these will flick the light switch, and once that switch is flipped, then a series of events always happens. What are the ultimate three major outcomes of turning on the complement system? That's right. The process of inflammation is initiated, opsonization, and cytolysis. So let's look at each of these components of the complement system, starting with the guts of that light switch, the core cascade. Here we see a nice graphic of which things are involved in the core cascade and then ultimately what they lead to. So the first of these is the protein C3. This is like the plastic part that you have to click, right? When you flick this, all of the rest of the stuff happens. You aren't aware of all this behind the scenes stuff, but you do know that you have to turn on the light switch. So activating C3 is a necessary first step, and activation involves breaking it into two pieces. One of those pieces, called C3A, goes on to participate in inflammation, whereas C3b participates in opsonization. And also this molecule activates C5, breaks it in half. C5a goes and works with C3a to promote inflammation, and C5b organizes these C6, 7, 8, and 9 proteins into a channel that pokes holes in membranes. So that's the light switch. What is it that can turn on the light switch? Well, there's the classical pathway. And what's the first event in the classical pathway? What's sort of the most upstream thing that has to happen first in the classical pathway? Yes. So the classical pathway is this one of many examples where adaptive and innate immune responses talk to each other. The initiating event in the classical pathway is actually something that happens due to the adaptive immune response. Adaptive immune response produces antibodies against specific antigens. Binding of the antibody to its target is a signal to start the complement. And it involves several proteins that we haven't talked about yet. So if we look at this picture more closely, remember what I told you is that there are 9 complement proteins, C1 through C9. But if we look at what's involved in the core cascade, we actually don't see all 9 proteins represented. What is missing? Yes, C1 is missing, C2 is missing, and what is the other one? And C4, that's right. And guess what? They all show up here. So the first thing that happens is whatever these antibodies are specific for, let's suppose it's a capsule polysaccharide of this bacterium, or could be a viral spike, a protein spike off of a virus. Whatever this microbe is, it's got antigens, the antibodies bind the antigens, and this is a signal to activate C1. C1 activates C2 and C4 by breaking them in half, and one piece of each of these activated proteins is gets together to create something called C3 convertase, which binds to and activates, chops in half C3. And now we have C3A, which can participate in inflammation, and C3B, which can participate in opsonization and initiate cytolysis through activating C5. The classical pathway involves an adaptive immune response as its initial trigger. the antibodies binding to their targets. Now, the alternative pathway doesn't involve the adaptive immune response at all. There's no antibody. It bypasses actually C1, C2, and C4. There is, however, a pattern recognition that has to happen. This pathway is tuned to recognize general lipid polysaccharide signatures on bacteria. So bacteria have a lot of, especially gram negatives, but gram positives too. They have lipid polysaccharide complexes that are distinctive to bacteria and not found on human cells. What are the proteins that do recognize those? What are the guys that recognize that pattern, lipid polysaccharide pattern on bacteria? It help to initiate the complement. If it's not C1, C2, or C4, and if the antibodies are not involved, there are 3 proteins. We haven't talked about them yet. What are they? If you don't remember, put a question mark. If you do remember, put it into the chat. I will give you a hint that each of these 3 proteins is designated by a single letter of the alphabet. So you could just put up 3 letters and hope for the best. That's right, Tao. It's factor B, D, and P. So here are these factors that can bind to these lipid carbohydrate complexes that are typical of bacteria. And these together directly activate C3, chop it in half, causing the whole complement activation process to begin. So we get cytolysis, opsonization, and inflammation. Finally, we come to the lectin pathway. Lectins made by the liver bind to another thing that's found on the surface of bacteria and that's not typical of human cells. And what is it? What do they bind to? What specific sugar? It is a sugar. It's not just all sugars. It's a specific one. That's right. Mannose containing carbohydrates. So there's the lectin made by the liver binding to mannose. This bypasses C1 because there's no antibody antigen, but it goes directly to C2, C4, activating them, and this becomes C3 convertase, activates C3, and then we get all of the things: inflammation, opsonization, cytolysis. We want to take a quick, more detailed look at the effects of complement activation. When we talk about inflammation, what are the C proteins directly involved in inflammation? We talked about this when we talked about the central cascade. Let's see if we remember. Yeah, C3A and C5A. If you have difficulty remembering which half of C3 and C5 is involved, C5A, C3A, A is for inflammation, okay? Opsonization, and on the other hand, involves C3b. And then cytolysis involves C3b activating what? That's right, C5. So that creates C5b, which organizes C6, 7, 8, and 9. Okay. Let's have a closer look at how this works. So these are histamine releasing mast cells that have cellular receptors for C5a and C3a. When that binding event occurs, it triggers the release of histamine containing pouches. This is a pro-inflammatory cytokine. And then C5a also additionally acts as a chemoattractant for phagocytes like neutrophils and macrophages. C3b, we could talk in more detail about, but let's just suffice it to say that it helps to coat the surface of targets like bacteria, viruses, and toxins so that phagocytes can find them. So this opsonization is like a coating flag process to make it easier for phagocytes to find the things they're supposed to get rid of. And cytolysis, C5b organizes C6, 7, 8, and 9 into a hole, punches a hole in the membrane, not just once, but over and over. Like that is to say, there's lots of these complexes, and so they are poking lots of holes. And a cell can go from being intact to being basically exploded in a short order. All right, so let's postulate a person who has a genetic disorder in complement proteins. Which of the following genetic lesions would cause the worst condition if you had a genetic mutation that disrupted C1, C3, C5, or C9? Which would be the worst outcome? Yeah. So put in your guesses. You only have 4 options. So no slacking. Even if you're not sure, just put something in. Remember, there's no points. There's no stakes. I'm not going to remember if you got it right or wrong. Great. That's right. I agree with all y'all. It is C3. And let's think about why that is. C3 is absolutely critical. It's the light switch. If you remove the light switch, you cannot turn on, even if all the guts are still there, and even if all the triggers are still there, you can't turn it on. So C3 would have the most devastating impact. It would cause the complement activation to basically stop altogether for any reason. Let's look at some of these other ones and what they would do. So if the problem was with C1, would you have a complete disruption of complement activation? And if not, what would be disrupted? What would happen if you lost C1? Yes, you would lose the ability to turn on the complement as part of the classical pathway, but you could still defend yourself against bacteria because they're likely to initiate the complement due to lipid carbohydrate complexes or mannose containing polysaccharides. And in those instances, the complement would come with full force. Now, what if we just disrupted C5? Would you still be able to activate complement? And what would the outcome be? What would be affected by the loss of C5? You would lose cytolysis for sure. And it would probably also blunt, yes, Haley, it would blunt your inflammation response. C3a would still be there, but C5a wouldn't. So you'd have less inflammation, you'd have no chemoattractant. And what about C9? Let's say we just get rid of C9. What's the outcome? What's disrupted here? Yeah, you would get no cytolysis, but you would still be able to activate the complement for any reason, classical alternative lectin pathway, and you would still get inflammation, and you would still get opsonization, but what you wouldn't get is cytolysis. Okay, let's turn our attention to interferons. Interferons are annoying. There are 3 of them. 2 of them do the same thing. 1 does something completely different. Let's find out. First of all, what are those 3 interferons? Name them for the chat. That's right. alpha, beta, and gamma. All right, so alpha and beta basically do the same thing. If you were to describe their function, what would you say? Put that into the chat. How would you describe what alpha and beta do? That's right. These two interferons are produced when a cell is already infected with a virus. And they're secreted locally, and they go basically door to door, knocking on all the neighboring cells to warn any cells that have not yet been infected to protect themselves. On the other hand, interferon gamma does something quite different. It's produced by the adaptive immune response in order to increase the phagocytic ability of macrophages and neutrophils. So let's consider how alpha and beta interferon function. So here's a cell that's been infected with a virus. The viral RNA is a trigger to make alpha and beta interferon. These are secreted and they go to neighboring cells. If they have not yet been infected, this gives them time, because it's a signal, to make antiviral proteins, primarily things that would inhibit, for example, viral polymerases, like RNA-dependent RNA polymerase. And in this way, cells that have not yet been infected can protect themselves from that first initial replication event. And once the virus reaches them, it can stop it in its tracks. Okay. From these options, what would you say is the best definition for innate immunity? Again, there's 4 options. Place your bets. If you're not sure, make a guess anyway. All right. From my perspective, this is the best answer. It's clearly not something that requires memory. It doesn't learn, and it's not that specific. This is a little bit too general. It doesn't protect us against all trauma and disease. However, it is some particular defenses present in all of us from the time that we're born. Smoking paralyzes the ciliary lining, your upper respiratory tract. Right? This is similar to the effect that anesthesia has, but it's like you're being anesthetized all the time. What effect will this have on your immune system? Select your answer. Again, if you're not sure, just take a guess. I would agree. So cilia are part of the ciliary escalator, so we don't get our pathogens removed from the airway. That's going to put us at risk of a lot of respiratory diseases. And the ciliary escalator is definitely part of our innate immune response. So A and C is the best answer. Let's go through each of these and decide if they are antiviral. Okay, so these are a bunch of defenses that you have in your innate immune response. Let's start with cytolysis, which is one of the outcomes of the complement system. Is this antiviral, yes or no? And again, you need to weigh in. If you're not sure, 50-50 shot being right. Great. So the correct answer, as many of you have discovered, is no. If you can explain why the answer is no, and why you chose that correct answer, go ahead and raise your hand. Why is cytolysis ineffective against viruses? Let me just ask a few of you from the chat. And if you don't have a good reason and you just guessed, that's a perfectly fine thing to say. But Nikki, can you tell us why you chose no for cytolysis is not antiviral? That is correct, but what was your reasoning? Are you there, Nikki? Are you able to tell us? How about Haley? Oh, Nikki's gonna pass. Yeah. Haley, do you have an answer to this? You had a question mark, but do you have a guess as to why?
Sorry, did you say Haley?
I did.
I think I'll pass. My question mark should have just been a question mark, not even
Okay.
no.
But you were right, as it turns out. Amy, what about you? What do you think? Are you able to unmute and tell us, Amy? Anyone else? What was your reason for choosing no for cytolysis? Let's think about what this actually does, right? Pokes holes in membranes. Viruses have no membranes. That's right, Tao, exactly right. So this is pokes hole in cellular membranes, which viruses don't have. And yes, they have an envelope, but it's not required for them to stay intact. Transferrin, yes or no, antiviral? Transferrin. Put in your guesses. Don't be afraid to be wrong. Don't be afraid. Don't overthink it. Just what do you think? Yes or no? Keep those guesses coming, guys. Okay, so transferrin is also not antiviral. Raise your hand if you can explain why. Thinking about what transferrin does, why it's important, how it acts. Why is it not antiviral? Because it isn't. What's the reason for that? Let's go to Dorothy. Can you tell us why you chose no? And if you're not sure, that's fine. But if you do have a reason, tell us what it was. Okay, that was a guess on her part. Janelle, do you have an explanation? Also not sure. Tau says because it binds iron, which is true. Virus does not produce iron binding matter. Yeah, so that is on the right track. Let's think about what iron is used for by bacteria. We use transferrin to keep iron away from bacteria. We need it for aerobic respiration. We're also trying to prevent bacteria from getting hold of it because they could use it in their aerobic respiration. Viruses don't do any metabolism, right? What they do is they're machines designed to get their genetic material into your cells and then get your cells to do the metabolism, but they themselves are not affected by this. Lysozyme, think about what lysozyme does. Is it antiviral, yes or no? Great. If you have an explanation as to why the answer is no, because it is no again, raise your hand or write it into the chat. Why isn't lysozyme antiviral? Thinking about what it does. What does lysozyme target? Why is it not antiviral? That's right, Kelly. The target of lysozyme is peptidoglycan, which is present in bacterial cell walls, a structure that viruses do not have. Okay, what about interferon alpha? Antiviral, yes or no? That's right. Interferon alpha, just like its friend interferon beta, is antiviral. And how does it work? Raise your hand or write your answer into the chat. How does it work to be antiviral? It's secreted by virally infected cells to warn their neighbors to make enzymes that stop viral replication. Good. Last but not least, we have interferon gamma. Not really antiviral per se, typically used to increase phagocytosis against bacteria, but you could imagine it could be sort of collaterally antiviral. Okay, one of the things I asked you about in the beginning to pay attention to was that even though the innate and adaptive immune responses are separate, and even though we haven't yet covered the adaptive immune response, at least not in my class, they nonetheless talk to each other. Can you think of any examples from what we talked about today, or that you know about what we didn't talk about today, that are examples of the ways in which these two branches of your immune system collaborate and communicate? Put those into the chat, anything you can think of. Yes, Kelly is right. Complement system. Can you say more about that? Or can anyone else kind of add to her great answer? The complement is part of one such example. And are there other examples that you can think of? Well, there's more examples than what I'm going to show now, but it gives us a taste, and we're going to sort of see some more of these next time. Kelly sort of hinted at the first of these, which is that when the complement is activated by the classical pathway, that's one of these iconic examples of way in which the adaptive immune response calls on, leans into, asks for help from the innate immune system. We also talked about, especially you saw that in the video, how phagocytes follow chemoattractants, but then they're able to identify what specific things they should be ingesting because they're coated with antibodies. Antibodies are produced by the adaptive immune response, but they allow phagocytes, which are part of the innate immune response, to do their job. And some of you also are aware that T helper cells, adaptive, activate macrophages, innate. There's other things too, right? Like the dendritic cells talking to T cells, etc. Okay, we do not have enough time to play our 20 question pathogenesis epidemiology Kahoot again, but, and we probably won't have time tomorrow, but we will do it as part of our course review. This is not quite accurate. I apologize. Let me just, oops, fix there we go. So you actually have Tuesday night is your pre-lab 8 deadline, and Tuesday is supposed to be here, and this is supposed to be here. Tuesday is an important day for a couple of reasons. It's our last lecture, basically, or our last Zoom together, and we're going to talk about the adaptive immune response, so we want to prepare for that. And then the other thing about Tuesday is that if any of you have not yet submitted your lab report, I actually am still accepting them until Tuesday night. Furthermore, if you felt rushed to get the lab report in on Sunday, and then afterwards you were like, oh, there's a typo, or I made a mistake, or I didn't get a chance to do as much work on the research section as I wanted to, then you are allowed to submit up to Tuesday a follow-up. However, if you've already submitted and you plan to resubmit, you must go in tonight and write me a note saying, I will submit later, Monday or Tuesday, because I'm going to start grading tomorrow, and I will do it based on what I'm seeing. So I always grade the most recent submission. So if you don't want me to grade you yet because you want to resubmit something, then let me know today. Okay, you'll just write a little note into that Dropbox. Otherwise, tomorrow morning, I'm going to start grading whatever I see, and I will continue to accept lab reports until the end of day Tuesday. So prepare for Tuesday's adaptive immune response Zoom. Wednesday's our last in-person lab. It's a lot of fun. Sunday, that lab 8 stuff from this week's lab will be due. And then Monday, we have our final Zoom meeting together, which is a Unit 3 and course review. Bring your questions to that meeting, and we will do the Kahoot as well. With that, I will stop the recording and take any questions that you have.